The idea for this is very simple, and it was born out of a challenge that DARPA gave me a number of years ago. I don't know if you know what DARPA is. It's the Defense Advanced Project Research Agency. Those are the guys that invented the stealth technology. They were actually the people that invented the internet. It was called ARPANET when they first invented it. So they gave me a challenge, and I won't tell you what it was, but the idea I came up with is you could make drugs, effective drugs, much simpler than we were trying to do it. And the idea was quite simple. If this is a protein, think about this as a protein. Most drugs are aimed at these pockets. They have to, because they need to have that chemical stabilization to sit in a pocket like this. Most antibodies tend to see these knobs, because that's easier for them to recognize. But that leaves about 80% of the protein surface that isn't being addressed by small molecule drugs or biologics, antibodies. So I think the solution to that was relatively simple. If you find a unstructured peptide, for instance, that would just lay in these canyons on there's shallow canyons. If you look at the structure of the protein, there's shallow canyons on the surface. And if you just found an unstructured peptide that was lay in this area and would make 2 or 3 contacts in there, and then find another peptide that would lay in this area that makes 2 or 3 contacts, even though the affinity of each one of these peptides might only be micromolar, all you have to do is link them together, and you create an affinity reagent with very high specificity and very high affinity. It's that simple. So, and the prediction would be, if you go through the numbers, it should be very easy to find these molecules. And indeed it is. And I'll show you how we do it. But the basic idea is that you just have a peptide that you find that binds one area, a peptide that binds the other area, you link them together. It's relatively unconstrained for the linking because these are loose peptides. And then you get high affinity, high specificity reagents. And they can be to proteins like AKT1 or TNF-alpha, or I'll show you they can also be to bacteria or viruses. It doesn't make any difference. So the process is this. We take whatever you want to have as a target, and you put it on a peptide array. And it's important that these peptides are not from life space. They're not from natural proteins. They're from random space. And then you put your protein on there, and you look to see which peptides bind that protein. And you make those as a collection. You can improve their affinity by a simple mutagenesis experiment because they're linear. Any improvement is additive. You just add up the delta G's, and it comes out in the end. And then you link them together, and you have your high affinity, high specificity reagent. They can be made simply and cheaply on large scale. That's all there is to it. Is that clear? It's simple, right? We've applied that to proteins, but lately we've tried to use this platform to address a really big issue, and that's antibiotic resistance. I'm sure all of you have heard about the prices of antibiotics in the United States. A lot of, there's about 23,000 deaths a year from bacteria that are antibiotic resistant. WHO and the CDC have come out saying that we could enter an era of post-antibiotics because we're losing the antibiotics we have faster than we're making them. And so, and if you look at the history of this, the part of the problem here is that we've been introducing antibiotics all along, although the rate of introduction has slowed down here. But if these are colored by classes, we basically are using the same classes of antibiotics over and over again. In fact, the last 3 introductions of antibiotics were derivatives of existing antibiotics. So the pipeline is really squeezing down, yet the loss of antibiotics is increasing. And this is from a Nature article, And then what you can see is that, antibiotics are delivered, resistance identified, the antibiotics are often still used for long periods of time after the resistances occurred, sort of ensuring that they'll keep the resistance around. But now the timelines have started to shorten up, and we see the resistance occurring faster and faster. So this really is not a good situation. And I think the problem is that we've gone at drug discovery for antibiotics in a very traditional ad hoc way. Each time we start to try and make an antibiotic, it's starting to do the wheel all over again. And we don't have a systematic way to make antibiotics. And there's always going to be resistance. Antibiotics are always going to induce a resistance. They'll always have a half-life. So we need to have a program to make antibiotics quickly and systematically. So we decided to see if we could take the SynBody technology to address this issue. And so now instead of putting a protein on there, we put a bacteria on there. And the bacteria has two dyes in it. One dye is a vital dye that it only stays inside if it's alive. And the other dye is a non-vital dye, so we can just see where the bacteria are. And that way we can pick out peptides that either just bind, if they just bind the bacteria, or peptides that actually bind and kill the bacteria. We can see those, so we can select either one. So we do that, and we get lead peptides, we improve, I'll show you, we can improve their activity in a simple method, and then we get this antibacterial SynBody. Here's the interesting part, is that most people use peptides from life space. That's the general concept. And I've argued for a long time that's really not a good idea. Because life space only occupies, think about this as the space of all 12 weeks. That's about 10 to the 19th 12 person in this space. Life space occupies this very, very small part of this. Because life is very constrained. It only occupies a small part of the protein space. So that means there's all this space out there that we could be using. So what we look for in that peptide array is that we go to all of peptide space and we sample it evenly as represented on this array. And that gives us a very powerful diversity of binding capabilities. Is that clear? Is that understandable? Okay. By the way, if you have any questions or comments, don't bother. Just interrupt me. That's fine. So this is an example of the kind of things we do. So we put the bacteria on there. This is one spot on the array of 10,000 peptides in this case. There's 10,000 peptides on the array. We take a sample from 10 to the 19th space. And then here's the bacteria that are binding. In this case, these bacteria are binding and they're alive. In other cases, we see with a different dye, we will see they're binding and dead. And so we can distinguish which peptide is which. And so then, if this is, I don't know if you can see this, but there's the bacterial colonies on here. Here's the negative control peptide. You can see all these colonies. These are the binding peptides, and it just doesn't do anything for them. It just binds. And this is a lactic peptide. There's no bacteria on that. So that's the way this works. And you can see that, you can't quite see this, but this is the growth with the negative control. This is if we add the binding peptide, it doesn't have much, I'm sorry, this is actually the negative control. If we add the binding peptide, it actually stimulates the growth for some reason we don't understand. But if you add the lactic peptide, it does inhibit it, but if you put them together, you can't see this, but there's a line down here that it kills them all. So now using peptides for antibacterials is not a new idea. It's been around for a long time, but it hasn't been successful. Most of the peptides that have been used are the natural peptides. You yourself have a huge number of peptides that are produced in you as antibacterials. Almost every mammalian, every system, every vertebrate makes antibacterial peptides. And those have been the source of trying to make pharmaceutical peptides, but it has not succeeded for a number of reasons. One is that they have low activity and relatively high toxicity when you try to use them systemically. So you have to have these high doses. Oh, thank you very much. You have to have these high doses. They have poor protease stability, so they degrade fast, and they've been expensive because to make these peptides. I should say, though, the cost of peptides has gone down 8-fold in the last 3 years. So the inhibition from cost is really not there anymore for peptides. But what about these other two issues? One is that we can pretty simply, that's nice. Looks nice. We can pretty simply modify our peptides to change the performance and make them more specific. And if by this simple protocol, if this is the sequence of the original peptide, all we need to do is make a plate of alternate peptides where we substitute each amino acid at this position, each amino acid at this position, and then assay them for activity. And then you can see, you can pick out the alterations that make them much better. And all you have to do is add those up and you get a much better. You just make a peptide that has this change, and this change, and it becomes a much better peptide. You can improve it 1,000-fold in its toxicity. You can also make it very selective. So you can ask for, because all this is in vitro, you can ask for a peptide that kills aureus but doesn't kill Pseudomonas. So if you wanted to, in the future, we may well have antibiotics that are very specific. They only kill the bug that you want to kill. It doesn't kill the broad spectrum, which is probably good because there's increasing evidence that all these other bugs are doing you good. You don't want to kill off every bug in you. You only want to kill off the bad guys. So we've shown that you actually can make a drive specificity to one bug against another one. And then you can also reduce the toxicity because you can just screen in a hemolysis assay whether it's less hemolytic or not here. So, and this is just an example that we can do that. We can use that technology to, yeah.
If you superimpose those data matrices, do you see any correlation?
Of which?
Like previous slide.
Yeah.
So now, if you take the first and the second one and you superimpose them, will you see any correlation between those?
Not once in a while you do, but not. It's not 1-to-1. So making something more toxic against one bug does not necessarily make it less toxic to the other. There is some correspondence, but it's not 1-to-1. And the same thing with the hemolytic. Something that makes it more toxic does not necessarily make it, to the bug, does not necessarily make it more hemolytic. Good question. So this is just an example where we're looking at MRSA, of antibiotic resistant and non-antibiotic resistant. As you might expect, it doesn't make much difference. If you're doing something to the membrane, it doesn't make much difference to whether it's antibiotic resistant or not. And you kill both of them. And you can increase, by that kind of mutagenesis method, you can increase the, you can see it's not effective against Pseudomonas anymore, but it's quite, there's no hemolytic, and it has a very good therapeutic index by going through this simple procedure. Stability, that's relatively easy to take care of because you just substitute the amino acids in there, and it usually makes them much more stable for activity. So that same SynBody, we can get the half-life up to 240 minutes by substituting the amino acids for the L amino acids. So that's pretty simple. I won't go through this, but basically there's a screen to see if it's actually targeting membranes, and most of the SynBodies that we pick, because we pick the things that are lytic, are actually hitting the membrane. Does it work in vivo? We have some evidence that does. This is a MRSA. It's kind of an artificial model, but it is an in vivo model for MRSA resistance, and this actually is the best molecule we've seen that they've seen in working in this this particular SynBody in this model. So it does look good. I'm going through this relatively quickly, but I just wanted to show you that here's a simple process, a platform process for making antibiotics that, and it's easy to screen. It's much easier than dealing with small molecules. And it's not limited to bacteria. Here's, we can also put a virus on there, and in this case, flu, and the virus can be killed. It doesn't make any difference whether it's killed or alive. For virus, we took out the peptides, make SynBody. We've worked out the chemistry here where we can make a lot of combinations very quickly and measure their activity. We've done this for flu. Here's how we pick them out. We look for peptides that bind both live flu and UV inactivated flu, and you can see that there's a number of them that have high affinity for that. And then we make the combinations of these peptides together, and in every mix we get both the homodimers and the heterodimers. And they're in a 1-to-1 ratio of homo to heterodimers. We do an HPLC purification. It's easy to pull them out because there's enough difference between the arms that you can purify them. We do this quickly, and this is in two types of assays for the effect of the SynBody on flu. The cytopathic production assay. These are two different SynBodies that we have here. This is the estimated IC50 for these. There's also a plaque reduction assay, also an in vitro assay, and you see the SynBody works reasonably well in a plaque reduction assay. It's stable in vivo, just putting in a mouse serum, and it is not toxic. This is a toxicity assay in mice. And it seems to have some efficacy in allowing the mice to survive from flu. This is a flu challenge. Mice that were mock protected, and these are mice that got the flu virus. About half of them survived. So it can work as an antiviral also. So I'm not going to go through the data on the specifics of the target. I just wanted to show you one cool thing. So DARPA challenged us to say, well, what if an unknown pathogen came along and we needed to make an antibiotic, 10,000 doses of an antibiotic or anti-infective in 1 week? Could you do it? And so we came up with this scheme to do this that actually worked. So what we did is that we screened a whole bunch of different pathogens on this 10,000 peptide array, and then asked, okay, what were the 100 peptides that would cover all of pathogen space? That any pathogen would bind 2 or more peptides when we tested it. And then we made that 100 peptide array, but we also made a stock of grams of that peptide. Remember, the cost has gone way down, so it's easy to make grams of this material on each of the 100. So when we are given an unknown bug, we put it on our 100 peptide array, pick 2 that are binding that particular pathogen, put them together, and then we have stocks of this material so we can make 10,000 doses of this in 1 week. And then in the process, we also test them, even in 1 week, we can test them in mice for toxicity, we can test them in cell cultures for mutualistic activity, and so we can do this in 1 week. It's actually pretty cool that you can do that kind of technology.
Sorry, did you choose the array only based on those 20 pathogens?
Yeah, actually, we actually ended up using 34, but we tried to sample gram positives, gram negatives, RNA viruses, DNA viruses, because we didn't know you don't know what you're going to get, right? And so we just threw them all on there individually, and then it was a fairly complicated sort of algorithm for picking what are the best representation of that space, but we could get it down to 100. And it included peptides that were lytic and peptides that were binding for the bacteria. So it was a simple idea, but it actually worked. So, okay, so that's the SynBody technology. We have a core, that doesn't show very well, does it? We have a core if you want to use these arrays for this or the other purpose that I'll show you about later. I'll show you later how this works. So,
Stefan, I actually have a question. But can those bacterial viruses become resistant to SynBody?
We don't know. That's an interesting question. We think that it's much less likely that they'll be resistant because they're binding to the surface, let's say the bacteria, they're binding to the surface of membrane, and those types of antibiotics are harder to become resistant to. You can, but it's much more difficult. And it turned out that the one for flu actually was binding the nucleoprotein, so which is a conserved protein. I mean, that was just an accident, but it did that. So, is that okay? But that was just a warm up. Can I show you an even cooler technology? Can I get that other? I have another slideshow on there. Can you close that one? And I can't get over there. Can you close that? Yeah. And then you see that the other one says it's on the desktop. It says IMS. Can you close that one? Yeah. You can open the other one. Yeah. Okay, so that was about therapeutics. Let me tell you another one that's for diagnosis, and it's really simple idea again. And again, this is me. Now I'm representing both my center, HealthTel, a company I started, and the Russian-American Anti-Cancer Center that Andre heads up, that we helped start in Altai. So this is this technology here. We always, when we invent something, we actually try to do platform technology, solve the problem, something that can be broadly applicable. And the way we start is we start with the problem. What is the problem we want to solve? And then we see if we can invent something to solve the problem. So it's a little backwards from what most people do. And so the problem we want to solve is healthcare. How do we revolutionize how we handle healthcare? And we had two criteria for our inventions that we had to meet. One is we're going to solve healthcare, we're going to revolutionize healthcare, but we're going to do it for everybody, not just the developed countries. It has to apply to everybody. And a good case, an example of this, most of cancer occurs in the developing world. Most of the cancer and most of the cancer deaths occur in the developing world, not in the developed world. So if you really want to do something in healthcare that's going to have a big impact, everybody's got to be able to use it. And just to drive this home, for cancer at least, WHO is saying that there's an imminent disaster pending because the aging population, the infrastructures of developing countries can't handle the cancer disease. And the economic impact in developing countries is huge. It's almost $1 trillion in GDP costs just for cancer alone. So if we're going to do something, it's got to be for everybody. And if somebody says they're working in cancer and will only be working in developed countries, I don't take them too seriously about trying to really have an impact on cancer. So this is why I don't take this kind of stuff very seriously. How to cure cancer, that's nonsense. The cancer community does not know how to cure cancer. They have no clue. And all these guys that come out, like Stand Up to Cancer in the United States, there's this big thing getting all the Hollywood stars together that they're going to get enough money and a lot of chutzpah, and you're going to get cancer cured. And they have something like this. This is a quote. When the stars come together, cancer doesn't stand a chance. And what a bunch of bullshit. The cancer community is really stalled. It's not doing anything very effective, and it hasn't for a long time, and it's spent a lot of money not doing anything very effective. So, and this is their vision of the future, at least in the United States. How to cure cancer, positron emission therapy. It costs about $100 million to build one of these units, to put a cyclotron in the top of the building, you bring people in the bottom and zap them with positrons, and it costs $30,000 to zap, $30,000 US dollars to zap. So this is not a developing world solution for cancer, nor is personalized medicine. Personalized medicine, first of all, you get a tumor. You have to have a tumor first. Then you sequence your tumor, and then you get a drug. And those drugs often cause the course of treatment often cause in the United States is more than $100,000. That's kind of third world treatment for cancer. This is the way the cancer community going, in the U.S. at least, is just nonsense. It's a stalled, decrepit, useless community as far as I'm concerned. So, the other criteria, and this always gets me in trouble with all my oncologist friends, but I don't care. The lay community in the United States is just enraged with cancer treatment because of the lack of progress with all the money that they put in. The other criteria for this invention had to be, it had to allow the transition from post-symptomatic medicine to pre-symptomatic medicine. If you really want to transform medicine, you revolutionize it, you have to become pre-symptomatic. You have to detect early and treat early. So those are the two criteria that we met. So we listed the sort of engineering specifications to meet that criteria. One, it had to be, if we're going to invent something to solve that problem, it had to be comprehensive. It had to work for all diseases, cancer, heart disease, infectious disease. It had to work for everything. It had to be very sensitive because you want to have early detection. And so, but to do these two things, it had to be simple and inexpensive because healthy people had to use it. Healthy people aren't going to use something that's complicated or costly. And then it had to have high specificity because if you're going to detect a disease early, you have to know what to treat. So these are the criteria. This one in particular is really hard. And that's because of this blood dilution problem. The biomarker field has really deluded themselves to thinking that they can do early detection of disease with biomarkers. You can't. It just doesn't work. And here's a simple example. This is actually published by Sam Beers a couple of years ago, but we had worked this out years ago, and it was very obvious. If this is a tumor the size of an olive, let's say, and it's putting out biomarkers, it goes into 5 liters of blood. It's going to be diluted to such an extent that no technology that we have, every technology we have is at least 10,000-fold too insensitive to pick that up. So the biomarker community that for years was going around saying we're going to detect early cancer with biomarkers, it's just nonsense. It doesn't make any sense. And so it was a simple calculation that would not work. The only way you can do early detection is that you have an amplification system. You have to amplify that signal in some way. And there's basically 2 ways to do that. One is PCR, amplify nucleic acids that tumors put into the blood. The other way is to use the B cells. B cells are sort of natural bots. They're floating around in your system all the time. They're seeing what's going on. They know what's happening. And if they see something, a single B cell sees something going wrong and gets activated, it amplifies the signal. You can't see this. 10 to the 11th fold in 1 meter. So it's a natural amplification system of anything going wrong in the body. So the technology that, based on that concept, the technology we developed is called immune signatures. And it works like this. You take a drop of blood, you dilute it 1,000-fold into buffer, just standard buffer. You put it on a chip. It's a peptide chip, and I'll tell you about it in a minute. Put it on a peptide chip. Antibodies will bind to that peptide chip. You wash off the excess, you detect where the antibodies are, and you get a signature. Are you guys familiar with heat maps? You must be, right? It's just like genomics. You guys, how many people know how to read a heat map? Anybody? Okay. Let me walk this through because I'll show you some data and it's all in this format. So let me just tell you how it works. This is everybody that has a disease, and these are people that don't have the disease. Every column represents a single individual. And every line across is a single peptide. So this individual here had no reactivity with these peptides, but high reactivity with this peptide, while people without the disease didn't have the reactivity with this peptide. And reactivity meaning lots of antibodies. Lots of antibodies is red, no antibodies is blue. Is that clear? Is that okay with everybody? So to get that, it's all a piece of cake from here on. Okay, so the vision we have is that everybody on a regular basis will send in a drop of blood in the mail to a central analysis system. They'll analyze them for any condition that they may have, and then they'll get that information back on the web to either themselves or themselves and the physician. So here's the arrays that we have. They're all addressable. So each point down here is one peptide. And we know what that peptide is. We know its sequence, and it's always the same peptide in the same position. Then we throw the antibodies on, they're in a buffer, they bind. Turns out only antibodies bind to the surface, and that's because of this dilution. Then you wash those off, you come back and detect the antibodies with the labeled secondary antibody, and that's all there is. But it's a true signature. It's not looking for a specific biomarker, it's a signature with a fingerprint. And so, like I just said, we pick those peptides from random peptide space. We don't pick them from known sequence space. And that turns out very important. The most important aspect of it is the same chip, the same set of peptides can be used to diagnose anything. You don't change the chip at all to use it for a different disease or even for a different species. It's the same thing always because we're picking it from random space. It also has very high resolution. You can't see the bottom, but here we show that we only had to use 10 peptides off of that 10,000 peptide array to distinguish all of these monoclonals from each other. Every one can be signatured individually from 10 peptides. So it turns out that by using random space like this, you splay out the antibody binding specificity to a very high resolution. So from a single feature, you get the intensity, which represents how many antibodies are there. You can get isotypes. I don't know how many of you have immunology background, but there's different kinds of antibodies. And you know the amino acid sequence. And it's very sensitive. I won't go through this, but this is, because it's a true signature, this is how you have to establish a diagnosis, right? You take a bunch of people that don't have the disease of interest and a bunch of people that do, and you run each of one of their sera on the array, and then you look for a feature that's reproducibly different between the people that do and don't have the disease. And then you compile those peptides, and that becomes your signature. So it usually takes, let's say, 100 peptides that you define as your signature based on. This is called a training set. Then you go back to a blinded set. This happens to be ovarian cancer. You go back to a blinded set, and you say, okay, those 100 peptides, how accurate are they in distinguishing these samples from each other? How do I score? And in this case, it scored near perfectly, too, in terms of classification. It doesn't always do that well. So what's the feature? It's, again, the same chip is used for all diseases, detects any kind of antibody. Because it's antibodies, and antibodies are very stable, you can use sera that's 20 years old, that's been stored for 20 years old, and you can establish your diagnostic on that. You don't have to do a prognostic study. There's no sample preparation, it's very sensitive. I just wanted to check this out because not everybody gets this. If you take one drop of blood and put it on one chip, you can test it for any diseases you want. As long as you have the signature, they don't have to be related to each other. You can test them for an infection, diabetes, breast cancer, doesn't make any difference. So it's really quite amazing. I won't go over this, but we showed that you could send these things through the mail. Now, what I've been telling you about up till now is chips with things that are spotted arrays. We actually have stocks of 10,000 peptides that we had made, and we spot them with a spotter on these last slides. And that works fine, but it's not scalable. If we're going to do what I really said, we have to be able to make 7 billion of these chips a year, right? Because there's 7 billion people, and if you get mass sequence a year, that's a lot of chips. There's only 2 companies that make a lot of things the same on that scale: McDonald's and Intel. And so we chose to use Intel as our model. And so what we went to is a new way of making these chips. It's made on wafers. These are silicon wafers. And we use standard photolithography to synthesize peptides in situ on these silicon wafers. So we use a mass-based system. We shine the light through. The light activates a photoacid. The photoacid deprotects the amino acids right under that spot. When you add another amino acid, it adds onto there. And it's just like standard TBOC chemistry. There's nothing special about this chemistry. No special reagents or anything. It's Merrifield's chemistry. And so in this way, we do the cycles to build up the peptides in CG1 here. This is not a simple matter. I mean, working out that chemistry was not easy. And what was important is that we worked out a way to do the mass spec right on the spot. So we could do the mass spec on the spot and figure out what the nature of that peptide is. And that's really important because it tells you exactly what the chemistry is that you made, including if you didn't make it right, you know what you did wrong from the mass spec. Remarkably, we just published this in Nature Communications. The stepwise yields are very high for these amino acids, much higher than standard chemistry. I think this is maybe a surface chemistry effect. Part of the reason for going to this thing is not only so that we can scale up the numbers, but I'm sure you're probably aware of Moore's Law. Well, there's also the inverse of Moore's Law. That is, the more you make, the cheaper it gets. And we expect that we can make these chips for less than $10 a piece. This is what it physically looks like. This is a 20 centimeter wafer, silicon wafer. And then each one of these, I don't know if you can see, but each one of these little gray spots is an array of 350,000 peptides that we've synthesized as each year. So each one of these has the same set of peptides on it. And then, but we cut it out in the shape of a slide, a standard slide, so that those slides can go into standard machinery for processing, slide washers, et cetera, et cetera. So this is after we've done the immunofluorescence. This is what one chip looks like. That's a half a square centimeter. There's 350,000 peptides on there. Each one has a diameter of 8 microns. If you're curious, this is what one of those slides looks like. And because there's chrome in between the features, you can reflect off there. Each one of those is one of the features on the array. Yeah.
Sorry. You said that there are about 350,000 peptides on the chip. Are they random or are they customized?
No, they're customized in the sense that we chose them to represent evenly random space.
To represent all the diseases? All
No. There's nothing to do with the disease. We forget about
want to
disease.
see all the picture.
Yeah, all we did was just imagine that this is all possible peptides, right, of a certain length. All we did was forget about diseases, forget about anything to do with life space, any natural protein, forget about it. All we did was just we picked peptides off that surface so that they evenly were spaced in that, so they weren't clumped up, so that we now have an even representation of all of that space. Even though there's 10 to the 19th possibilities, we only have 350,000 peptides.
And you enroll healthy people, and in the beginning you enrolled healthy people, and now you look after their diseases if they have any, and compares patterns of
Yes, yeah. But we don't have to enroll them. Sometimes we do.
Enroll, of course.
Sometimes we do
Just
enroll. Yeah, most of the time these are samples that have already been collected.
How many years do you look after the patients? Not patients,
Yeah. So let me show you an example.
yes, for example.
Okay, so we started a company that's going to start doing this let me skip that. This is infectious disease, showing you how well it works. Let me give you an example here. These are breast cancer. This was a blinded test of women with breast cancer.
On which stage?
These stages are 2 and 3. This is 4, right here. And these are healthy women, so they're without cancer. And in the blinded test, it was about 95% accurate in picking the people with breast cancer from the people that did not have breast cancer. Most of these samples were collected at least 10 years ago. So they are old samples, and it doesn't matter. And in fact, in this case, these are 5, you can't see this, but this is 5 different types of brain cancer being distinguished here. Brain cancer is easy to distinguish, it turns out, by this method. But many of these samples were 20 years or older that we were using to do this diagnostic development. So the antibodies are unbelievably stable as long as you keep them as sera or blood. So that's easy to do. It's not limited to cancer or infectious disease. This is Alzheimer's disease, and looking at the ability to distinguish controls from age-matched people with Alzheimer's disease. This is not autopsy-confirmed Alzheimer's disease, so some of these people may actually have it and not know. But it was about 90% accurate in being able to distinguish Alzheimer's disease. That's better than the current clinical practice.
And it really allows for diagnostics before symptoms.
Well, these people had symptoms. They were diagnosed with Alzheimer's. But
It's Alzheimer's disease.
right. But we've done 2 things. In a mouse model of Alzheimer's, we've shown, and we published this, that we can pick up the earliest stages of Alzheimer's before there's any symptoms. We can see the signature for that.
Without any symptoms.
Without any symptoms. And we've started a small scale study of people in Colombia. There's a small community that has a high frequency of a mutation, so they developed Alzheimer's at 42, almost right on the dot.
And what about cancer?
Early? Yeah. So we've done, in mouse models, we've done breast cancer studies, and the same thing. In fact, it's easier to pick up cancer early than late. So it's easy to do. In pancreatic cancer, we've looked for PANITs, which is the early hyperplagic phase of pancreatic cancer, and we can see that signature. And it's different than the signature that people that have pancreatic disease. They have a different signature than the people that are just starting to get pancreatic disease.
You said that this technology must be cheap because it's for health people.
Yeah.
And what is the problem?
The cost?
Yes.
Well, right now we're doing this on a research scale. So the cost is probably, depending on who you are, if you're a company, we charge you more. If you're not, we charge you less. But it's on the order of $150 right now. But we want this to be down below $100 in the U.S.
And it depends on scale.
Exactly on the scale. And also remember, every time because it's the same chip, every time you add a disease, it doesn't cost you anymore. You don't change the production at all. You just add information. So if you send in, if we have the company's launching where they're going to test for the 4 major cancers, lung, breast, prostate, and colon. But if you add a new signature on there, it doesn't cost anything. If you want to say, oh, I'll also do Alzheimer's, just look for the signature. And you can even go back historically in somebody's signature. After you develop this signature in the future, you can go back to the data. You don't have to do the test again. The data's there, right? So we've done, and this is something we published in PNAS recently. This is looking at 15 different diseases in a blinded test. So can you distinguish 15 different diseases, most of them cancer, from the healthy people? And we were with varying accuracy, but most of them quite high specificity. We were able to distinguish all of them from each other. Now, that data is all on a chip with 10,000 peptides. We now are going to one with probably 125,000 peptides for the standard diagnosis we'll use commercially. But just to show you what the power is, this is distinguishing, I think, 7 different diseases, all infectious diseases, from each other. This, West Nile, the commercial product, cannot distinguish West Nile from dengue, but we can easily distinguish it. And we can also ask, how does the resolution map with the number of peptides you have? And what we find is that if we compare a chip with 10,000 peptides to 330,000 peptides, it's about 50 times better to use 330,000 than 10,000. So the more peptides you have, the more resolution you get, at least up to that level. So I think we can do all those things. I think we can beat the comprehensive, the sensitivity. I think it's possible to scale now that we're switching to that type of system. And I think this vision of everybody sending in a drop of blood on a regular basis for a comprehensive analysis of your health status is very reasonable to do. And that would revolutionize medicine if you could do that. So let me just give you one last example of how this might take place. So right now, this is the way we take care of cancer. People get a cancer, it can be there for years before it's actually picked up by imaging or symptoms. In the United States, this system leads to over 500,000 people dying per year, so it's not a very good system. And huge costs. We spend about $250 billion a year on cancer treatment in the United States. So imagine that you had this system where everybody's monitoring their health on a regular basis by sending in a drop of blood, right? So now you're monitoring and you pick up the earliest stage of this tumor when there's only a few cells here. And now what you do is you come in and get a systemic treatment. So for instance, you may have heard about these checkpoint inhibitors that people are using in the cancer field. They're very exciting. What they do is, when you have a cancer, as soon as there's a cancer, the immune system is reacting to it. It's trying to take care of it. It probably does, or most of the time. You are developing tumors all the time, and they're being taken care of. But sometimes the part of your immune system that suppresses the active part that's trying to kill the tumor, it's suppressed by the other part that's trying to regulate the tumor, the immune response. Well, what happens if you give people these checkpoint inhibitors, it knocks down the regulator and it lets go of your own immune system. It sort of effectively vaccinates you against your own tumor. And so now, what you envision, you have the first sign, you activate your own immune system, or with chemotherapy, you take care of this tumor when it's small and early before it's had a chance to evolve, and then you monitor to see that it disappears, and that's it. That would be the future of managing cancer. You never have symptoms, you never have to go into radiation, or wait till your tumor's big enough to get sequenced, or anything else like that. It would be much, much cheaper to do. So this should be the vision of the future of the cancer community. Unfortunately, it isn't. So it's hard to get these things done in people. It takes longer. So we're going to do it in dogs first. So we started another company, Calvary, to do all this stuff in dogs. So at least in the United States, your dogs will have better health care than you will earlier. So we probably will be able to prevent cancer in dogs before we can go to people. We'll see how it goes. So I won't go through all this. This is another. It's an interesting platform that you can do other things with. You don't have to just use it for diagnosis. There's other things you can use it for. So this is the summary. I think it is a universal diagnostic platform. It really is simple and sensitive, and it also can be employed as a discovery tool. Back to this, we have this core that you can send in samples, you can use it to make SynBodies or whatever else you're interested in, and it's a simple system to use. And I wanted to point out that I'm here because I started a collaboration with Altai University and the Barno Regional Cancer Centers to set up a core there to use this technology for their regional cancer center diagnosis. And Andre is heading up this core, and eventually that core will be available in Russia over there. And in fact, in relatively short period of time. And these are the people that helped me do all the work. In fact, they did all the work. I just want to give them credit. Any questions? Simple is good.
Very impressive.
Yeah.
But what's the problem? It's a wonderful technology, but now it doesn't work. What's the problem? What's the curve?
The problem? Yeah. Okay. So it's not that it doesn't work, but there is so the question you should ask is, what's the major risk, right? The major risk, I think, is the stability of the platform. It's one thing to do it on a research level and to make these things that you use in these smaller studies. But if you're going to use this on a regular basis and make millions or billions of these, you have to have something that is stably produced. It has to be like McDonald's. And so that is the major issue. How, not even the scaling and the numbers, it's the uniformity of the platform. That's the major challenge. Yeah.
Yeah. About a couple of years ago, Intel and Stanford actually launched something similar.
Yeah.
Same peptide chip. But is there like any difference, or your technology is just improvement of this? And why it's not yet to this scale where we can see it in a diagnostic lab?
Yeah. So we will soon for dogs. That's actually launched, and then probably the first product we'll have for people will be, ironically, not from the company. It'll be for valley fever. I didn't show you this technology. So Intel launched. This is an interesting story. So we did this independently. We didn't have anything to do with them initially. Intel actually launched an internal program to try and make peptide arrays, and they got to the point that they could do very rudimentary stuff, and that's what that Stanford report was. That's not anywhere near this scale. Like this. But the problem with that is they didn't know what to do with them. They didn't think of immunosignatures. That was my image. So Intel dropped the project because they saw no business for making peptide arrays. And of course we were driven by the business and what we saw that we could do from within in the first place. You know, we knew about immunosignatures, and we were developing the fabrication system in parallel. So we own all the patents now. So, yeah.
So they don't analyze the data?
Intel?
Yeah.
They never thought of using it to immunosignature. The idea, most people in the diagnostic community and the immunology community didn't think this would work. To take a drop of blood and profile antibodies, they thought that was a dumb idea. And there were biochemical reasons to think that was right. But I found that a lot of times it's good to, when everybody says that won't work or you should go this way, it's good just to go in the opposite direction, just for the hell of it. Because every once in a while, you discover something that nobody else would have. And so I just, in spite of everybody saying that this was not a good idea, and I agreed with them with the actual calculations, I went out and bought 10,000 peptides, which cost a lot of money, and learned how to print them and tried that experiment and it worked beautifully. I should point out, these antibodies are binding these peptides with micromolar affinity on the array. So they wouldn't stay there unless you put the peptides close enough together. So if they're close enough together, you get an avidity reaction. You guys know the difference between affinity and avidity? Right? Yes. Okay. So if you put them too far apart, more than 6 nanometers apart, this won't work. The whole system won't work. They have to be within 6 nanometers of each other in order to get enough avidity to hold them on there for the time you would wash them and look at them. The devil's in the details. Yeah. Sure.
From chemical point of view, once you synthesize different peptides, you synthesize the carbon. And I remember that the way that was built, that during the peptide synthesis, very often you have shorter peptides, longer peptides, additional amino acids, etc. So, if you synthesize, let's say, 300,000 different peptides, what is the probability that you have incomplete peptides and shorter peptides which will interfere?
Yeah.
What is the risk of that?
So, peptide synthesis isn't perfect. It looks like we're getting in excess of 99% stepwise yield. So for a short peptide, on these 350,000 or so, they're about 12 amino acids long. And that means that most of the product will be what you thought, what you intended to do. Because it's a true signature, if there's other peptides on there that aren't exactly what you thought, it doesn't matter, as long as you're reproducibly doing that. Because that's the beauty of a signature. And by the way, if anyone had been doing the calculations, you'd say, well, if they're 12 amino acids long and there are 20 amino acids, then you're going to have 240 mass, right? So 200 Intel, the biggest number of mass Intel currently uses is 18. And if we were going to make 12 mers, we'd have to have 240. So one of the interesting parts was how to write a program that would allow you to use the minimal number of mass to get the most number of representation of peptide sequence. So what we actually do are make 64 mass to make our 12 mers. And so we get is a distribution blend with a mean of 12.
Once we have peptides in this course, it's achieved, okay? It's possible or can we reuse it?
To reuse it? We haven't really explored that from a regulatory point of view. That raises a lot more problems if you try to reuse a piece of equipment. So we've tended to look more to just getting the price down rather than try to reuse them. I think there are schemes that you probably could do that, but we haven't explored it.
Usually it's blood sample, right?
We've used blood, saliva, and spinal fluid. And any place there's an antibody, we should be able to detect it. If you use saliva, the antibody concentration is lower, so you just have to dilute it less. But it works fine.
Usually in spinal fluid, concentration is very low.
Yeah, it is, but it's certainly doable. We've done that for some infectious diseases that we look at spinal fluids and it works fine.
And in case of blood, there is a lot of proteins. Do they interfere with the peptides?
No, the antibodies are protease resistant, so they're really tough. So that isn't an issue. No, we haven't had any. Remember, we're diluting the sample 1,000-fold, and so we've diluted that all those enzymes and everything else 1,000-fold, and so we only incubate it for an hour and then we wash it off. So it doesn't seem to be a difficulty. We've done it with protease inhibitors without big inhibitors.
How do you select those test samples out of the whole space of
Okay. So the first time we did it, when we ordered the 10,000 peptides, we sat around and tried to be smart. What would be the best peptides to use? We knew we didn't want to use peptides from natural space because then it's too specific, right? So we all had our own arguments and nobody could convince each other. So we just decided to use a random number generator. And so that's how we picked the first guy. So then, but after that, we started to get data of what was working, and then there is a slight bias to some amino acids, so we incorporated that into the, we've done over 40,000 different samples. We've done 40 different diseases, so we have quite a bit of data now. So we use that, but then we realized we really, just using a random number generator doesn't evenly sample space. So we've gone back and we wrote programs that basically maximizes the distance between the sequence distance between each peptide we pick. And we keep picking peptides till they have the maximum distance between them in sequence. And that's how we do it now. So it's a specific algorithm for it.
and like business-wise or maybe a regulatory-wise right now? Because, well, probably comparable to 23andMe, that started actually with kind of the same idea when you send the sample by mail, they analyze it and they send you back the report. But I mean, this year, was it last year, when there were lots of problems with 23andMe regulatory-wise, what's your stand?
Okay, so the only difference, the only resemblance between 23andMe and what I'm proposing is that they send in a sample.
Okay.
The rest of it doesn't resemble at all. This is going to be done in a CLIA lab, a certified laboratory for that product. So it goes to the same place, so it's all done in a very systematic way. And it's certified laboratory. They didn't do that. They were sending it out to somebody to sequence it, and they had no quality control over the sequences that they were running. And so that's why they ran into trouble. If they had established a CLIA laboratory and had standardized laboratory procedures for the sequencing and its quality control over its accuracy, they wouldn't have run into any problems. But so it sounds like sort of like 23andMe, and it actually has had a negative impact on what we're proposing because people made that simple connection. But it really isn't related at all.
So in the near future, we can actually try to use the service.
Yeah, unfortunately, you can't. The way we understand Russian law to work,
We can't
you can't send a
samples
sample out.
yeah.
But that's why we're established the court here, so that it can be done and this isn't the only country that is an issue with. Mexico, you can't do this either, and stuff like this. So we'll probably have to establish it. The beauty of it is, it really only takes two machines, a slide washer and an aerator, and that's it. Anything else? Well, thanks for staying late. Appreciate it. On the last day of summer. It's probably the last time you'll get summer weather for the rest of the year. So, thanks a lot.
Thank you.
Good.